student, this is the second lecture of unit 3rd, which having the following headings like de novo synthesis of fatty acids and biological significance of cholesterol, and what are the disorders which are associated with this cholesterol metabolism is also described in this lecture. So, firstly we have to start with the de novo synthesis of fatty acid. In the last lecture we have already discussed about the beta oxidation of fatty acids and ketone bodies formation and degradation of ketone bodies. So, de novo synthesis of fatty acids or palmitic acid. So, palmitic acid is specified in these slides because in our AKtU syllabus we are having palmitic acid as a reference molecule for the synthesis of fatty acid, because there are so many fatty acids present in our body and we have to discuss especially palmitic acid for the formation of this fatty acid. Okay, fine. So, what is de novo synthesis of fatty acid and how it formed or how it synthesized? So, it will start with the definition of de novo synthesis. So, what is de novo synthesis? It is a scientist de novo which discover this fatty acid synthesis. It is a series of reaction which hydrolyzes or involves the method of production of palmitate from acetyl coenzyme A. So, acetyl coenzyme A is a very important intermediate or compound in the body, because acetyl coenzyme A is also used in the Krebs cycle. It is also used in beta oxidation of fatty acid. It is synthesized 2 molecule in one beta oxidation reaction, and this acetyl coenzyme A is also useful or important for the formation of ketone bodies, and this acetyl coenzyme is also produced in the ketolysis. So, this acetyl coenzyme A work as a limiting factor for fatty acid synthesis, fatty acid oxidation, formation of ketone bodies and degradation or oxidation of fatty acid like ketolysis. So, this acetyl coenzyme A work as very important compound in fatty acid metabolism. So, here there is production of palmitate from acetyl coenzyme A, and what is the location of de novo synthesis? So, de novo synthesis occurs mainly in the adipose tissues. Adipose tissues is responsible for the formation of fat and kidney and liver. So, these three different organs are mainly responsible for the formation of de novo synthesis of fatty acids. Okay. Then, what are the stages? What are the stages which are basically responsible for the formation of this palmitic acid or fatty acid? So, there are 3 different stages. The first one is production of acetyl coenzyme A and NADPH. The second one is conversion of acetyl coenzyme A to malonyl coenzyme A, and the third one is reaction of fatty acid synthase complex. So, these 3 stages are basically responsible for the formation of palmitic acid from this acetyl coenzyme A. So, we will start with the first stage, production of acetyl coenzyme A and NADPH. So, how acetyl coenzyme A is produced here? So, acetyl coenzyme A is a main requirement for the production of fatty acid. This is a main compound. So, this is also known as limiting factor here for the formation of fatty acid. This acetyl coenzyme A produced by oxidation of pyruvate in mitochondrial matrix, and then it free fatty acids or there are so many amino acids are also present here to form ketone bodies, because this I have already cleared this thing that acetyl coenzyme is basically responsible for the formation of different amino acids, different ketone bodies, and it is also synthesized by fatty acid oxidation. So, this is work as limiting factor. So, this reaction, the reaction which is the production of acetyl coenzyme A is taking place in the mitochondria, and it is impermeable to which is work as impermeable to acetyl coenzyme A. So, it is it can transfer acetyl coenzyme A alternative arrangement is required for the formation of this fatty acid coenzyme A. So, when this acetyl coenzyme is condensed to oxaloacetate and form the citrate here, which is these all are important. If you have remembered the second unit where the we have discussed about the metabolism of carbohydrate, here acetyl coenzyme A, citrate, succinyl coenzyme A, they all are present as an intermediate, because in Krebs cycle acetyl coenzyme A is reacted to form carbon dioxide and water. So, this is the main this is also correlated with that Krebs cycle. So, here acetyl coenzyme A condensed to form oxaloacetate, which is the intermediate product of Krebs cycle, and it forms citrate here. This form citrate is easily out of the reaction here or matrix here to transported by mitochondrial cytosol or mitochondrial matrix series. This formed citrate is particularly produces undergoing, there are so many reactions, there are so many enzymes, there are so many enzymes required here for the formation of this this fatty acid here. So, this is the pathway, this is the general pathway. You have already seen that there are so many intermediate present here, which is also similar with the Krebs cycle. So, in pyruvate, which is also react in the Krebs cycle here. So, in mitochondrial matrix, when pyruvate is reacted and pyruvate is reacted with the pyruvate dehydrogenase here to form the acetyl coenzyme A. This acetyl coenzyme is not directly convert into the citrate here. This is reacted with the again different various steps to form citrate, and citrates with the help of citrate synthase to form citrate here. So, pyruvate is when reacted with the catalyzed by the pyruvate dehydrogenase here to form acetyl coenzyme A, or pyruvate carboxylase is also reacted with the pyruvate to form oxaloacetate here. This oxaloacetate and acetyl coenzyme A again reacted together with the help of citrate synthase enzyme. It is also, this reaction is also common in Krebs cycle, and this formed citrate is again come fast and react with the this pyruvate to form citrate is again reacted with acetyl coenzyme A with the help of coenzyme A here, and there is one ATP is used in this reaction to form oxaloacetate, and this oxaloacetate is when reacted with malate dehydrogenase to form malate here, and this is again reacted to form pyruvate. So, this is a common pathway, this is the common pathway with the Krebs cycle and this fatty acid synthesis. So, this is the main responsible reason or production of acetyl coenzyme A. So, when acetyl coenzyme A is produced, so this is also convert into the pyruvate, citrate and oxaloacetate, and after that the formation of acetyl coenzyme A, there are the various reactions of fatty acid which are required for the synthesis of fatty acid by the fatty acid synthase complex. So, these reactions of fatty acid synthase are catalyzed by the different multifunctional enzymes which are known as acid synthase complex, which are present in the mitochondrial matrix and helpful for the formation of this fatty acid. Each monomer, each single compound possesses the activity or activity of different enzyme, and acyl carrier protein ACP, which is acyl carrier protein, which is we have already discussed in the beta oxidation of fatty acid, where acyl group is reacted with the carnitine carrier system to transport acyl activated acyl group from cytosol to inner mitochondrial matrix, because inner mitochondrial matrix is impermeable for transport of fatty acids. So, in that reaction this acyl carnitine system is known as carnitine carrier system. So, this sequence of reactions are discussed below. Here conversion of acetyl coenzyme A to malonyl coenzyme A. firstly we have to focus on formation of acyl coenzyme A and NADPH, which is now conversion to acyl coenzyme A to malonyl coenzyme A. This acetyl coenzyme A undergoes carboxylation reaction to produce malonyl coenzyme A. It is ATP dependent reaction because one ATP is utilized in this reaction, and there is formation of this reaction required biotin also, because this reaction required carbon dioxide fixation here, because one carbon dioxide molecule is also used in this reaction for the formation of malonyl coenzyme A. So, here in this reaction, when acetyl coenzyme A in presence of carbon dioxide, and this reaction is catalyzed by acetyl coenzyme A carboxylase and in presence of biotin molecule also, and one ATP is also utilized in this reaction to form malonyl coenzyme A. This is a general pathway for the formation of palmitic acid or de novo synthesis of fatty acid. So, we know that acetyl coenzyme A is the starting compound here for the formation of palmitic acid, and this acetyl coenzyme A is when reacted or catalyzed by the acyl coenzyme A carboxylase enzyme to form malonyl coenzyme A, and this malonyl coenzyme A is when catalyzed by the malonyl acetyl transferase enzyme or transacylase enzyme to form malonyl acyl carrier protein ACP. This malonyl ACP is converted or transported from the cytosol to mitochondrial matrix, inner mitochondrial matrix, to form acetoacyl ACP, acetoacetyl acyl carrier protein with the help of 3-ketoacyl synthase enzyme. And when this acetoacyl ACP and when reacted with the 3-keto reductase enzyme to form 3-hydroxybutanoyl acyl carrier protein. This 3-hydroxybutanoyl coenzyme A or ACP is when reacted with the hydrotase enzyme to form but2-enoyl ACP, which this but2-enoyl ACP is converted into the butyryl coenzyme A or butyryl acyl carrier protein, which is transferred or reacted with the thioesterase enzyme to complete this reaction with the repetition of 7 different times, because we all knows that palmitic acid have the 8 carbon compound in their structure. So, this reaction is repeated 7 times for the formation of palmitic acid here, and this reaction is also catalyzed by the enoyl reductase enzyme, which is responsible for the formation of this palmitic acid from butyryl acyl carrier protein. This pathway de novo synthesis of fatty acid repeated it 7 times for the formation of palmitic acid here. So, this pathway is reversed or competitively reacted for 7 times to form palmitic acid. And after that this is the de novo synthesis of fatty acid is completed here with this pathway, and now we are shifted towards the cholesterol. And what is cholesterol here? We all knows what is cholesterol. Cholesterol is a steroid which is basically synthesized in our body, and this is the longest lipid or fatty acid which is having the 27 carbon containing compound, and it is very important in our body. In a limited extent or limited concentration, it shows highly biological importance to our body when the intake and utilization are in control, and when there the intake or utilization is not under control, then it is causing the different disorders to our body. So, this is the structure of cholesterol. This is having the cyclopentanoperhydrophenanthine nucleus. This is the having the 4 different ring structure here, A, B, C, D. There are 4 different rings. These all 4 rings are combined here, and after the C17 there are a side chain compound, a side chain is also present, which is showing the C27 carbon containing compound, and this is the structure of the cholesterol, and this cholesterol is very responsible for the formation of various membranes, cell membranes and different organs here. ठीक है, and this cholesterol and its derivative, these are very important, and they are showing the they are having the different important role in the cell membrane formation or constituents of cell membrane, and they are also showing their importance by the formation of various different steroid compounds like sex hormones, male and female sex hormone, and corticosteroids. So, this cholesterol having the very important characteristic in our body functioning. So, what are the biological significance of these cholesterol here? This cholesterol is the major component of the plasma membrane and formation of lipoprotein. There are plasma membrane and lipoproteins are synthesized by the help of this cholesterol. Cholesterol is a precursor for the formation of all the membranes, all the cell membranes in our body, and this is also maintain the cell membrane integrity. They are also having the different strength, it is showing the strength of our cell membrane integrity, and this cholesterol also modulate the membrane fluidity and over the different physiological temperature. If the temperature of our physiological system is varied from higher or lower condition, this cholesterol maintain the total temperature of our body, and it modulate the fluidity or fluidity of our cell membrane. So, this is very important for normal functioning of cell membranes. And this is cholesterol is also helpful for the precursor for the biosynthesis of different steroids, hormones, bile acids, and vitamin D. Vitamin D is a good antioxidant. It is also helpful in detoxification of body, and it is also helpful for the protection of arteries also. It is showing required for the nerve transmission also. So, the nerve transmission which is occurs in our body is also effectively by the cholesterol here, and this is a main constituent of gallstone. Gallstone is also synthesized by cholesterol. So, it is showing some negative effects also in our body. So, when how to convert cholesterol to bile acids here. So, cholesterol around 50% cholesterol is converted into the bile acids, or which is excreted by the feces in our body, which serves as precursor for the synthesis of steroid hormones, vitamin D, copronesterol, and cholesterol. So, bile acids are synthesized in our body from cholesterol in liver, and this carbon 24 containing compound having 2 or 3 hydroxyl group in steroid nucleus, and they are showing or they are having carbonyl group in their structure, and these what are bile acids? They are amphipathic in nature. They are having both polar and non-polar group in their structure, and they are having emulsifying agents in intestine. How this cholesterol is converted into the bile acid? The cholesterol is when reacted with the 7 alpha hydrolase enzyme to form 7 hydroxycholesterol. Here 1 ATP NADPH molecule is synthesized in this reaction, and this 7 hydroxycholesterol is again helpful for the formation of cholic acid, which is a type of bile acid, and chenodeoxycholic acid. These both are having bile acids, and when cholic acid is reacted with the glycine and taurine and to form glycocholic acid and taurocholic acid, which is also a type of bile acid, and when this glycocholic acid is reacted with the intestinal bacteria to form deoxycholic acid here, and chenocholic acid is when reacted with the this chenocholic acid is when reacted with the taurine or glycine here, this is converted into the toro or glycoquinodeoxycholic acid, which is when reacted with the intestinal bacteria here to form lithocholic acid. So, this is the pathway for the formation of different bile acid from cholesterol. So, and how vitamin D is synthesized by the cholesterol is also described here. So, D7-deoxycholesterol is main intermediate for the formation of vitamin D from cholesterol. In this reaction, when dehydrocholesterol is reacted in UV light to form pre-vitamin D3. In pre-vitamin D3, there is just removal of this bond from this reaction, and there is CH3 group is again present here, and this pre-vitamin D3 is again converted into the vitamin D3 cholecalciferol here, and there is saturation of this 6-membered ring here, and there is removal of this attachment of this ring here, and there is one CH2 group present here, and this vitamin D3 or cholecalciferol is again converted into the calcitriol 125 dihydroxy cholecalciferol here. This is this reaction, this structure is removed here, and there is formation of this calcitriol here, and 125 dihydroxy 1 and 25 dihydroxy cholecalciferol is synthesized from this cholesterol in this pathway. What are the disorders of lipid metabolism? If there is disturbance or there is not proper relation between the synthesis of cholesterol and utilization of the cholesterol, they may show some disorder. So, what are the disorders? The first one is hypercholesterolemia. It is very common disorder, and in this disorder, level of cholesterol in blood is higher than normal, because synthesis of cholesterol is normal and we have taken cholesterol by the diet also, but the utilization of cholesterol is not proper. So, there is so many deposition of cholesterol in our body, which is causing fat in our body, of which is known as hypercholesterolemia. Yani presence of cholesterol in our blood is higher. So, this is because of the elevated level of low density lipoprotein. There are so many lipoproteins, low density lipoprotein, high density lipoprotein, very low density lipoproteins, which are synthesized from the cholesterol, and in this condition high density lipoprotein is concentration is reduced in our body, but concentration of low density lipoprotein and very low density lipoprotein and chylomicrons are increased in blood, so that they are deposited in our blood cells or arteries, and they are increased level of triglycerides, and decreases the level of high density lipoproteins, and this cause the quantitative lipid abnormalities, because level of low density lipoprotein and cholesterol whenever increases or greater than 190 milligram per deciliter is known as hypercholesterolemia, and this hypercholesterolemia is the starting stage for the abnormality of cardiovascular system, blood pressure. So, this is a crucial condition ki whenever we have to control our lipid concentration or cholesterol concentration in the blood. So, this is the main hypercholesterolemia. The second thing is atherosclerosis. This is the most scary stage when there is a concentration of cholesterol is again increases in the blood. They may cause atherosclerosis. What is atherosclerosis? Deposition of fat in the arteries. In our arteries there is flow of blood is common, and blood is commonly flow, and when there is increase in the cholesterol LDL and VLDL is increased in the blood, they may deposited into the inner layer of the arteries. Whenever deposition of fatty material in the arteries, they may cause plaque, or they may reduces the size of arteries, so that blood flow or blood have lowest space or size for the flowing of blood. So, this may cause blood increases blood pressure. And fatty liver, whenever there is excess buildup of fat in the liver, and this may cause accumulation of fat in the liver, leads to inflammation. There are two types of non-alcoholic fatty acid diseases which is having buildup of fat in the lipid, and this is basically not related to the alcohol consumption, because they are not related to the alcohol consumption. This is a non-alcoholic fatty acid disease. This is simple fatty acid fatty liver disease. Here buildup of fat in the liver, but they are not showing any damage in the liver. They are not showing any damage to the cell of the liver. And non-alcoholic alcoholic steatohepatitis, there is accumulation in the fat in the liver, accumulation of fat in the liver, but they are not showing any damage to the liver cells. In the reasons or causes are fibrosis, cirrhosis, or liver cancer. And the second one is alcoholic fatty acid fatty liver disease. This is mainly responsible by the alcohol. Those patients who are taking alcohol in more than a normal condition, they are having the alcoholic fatty acid disease. That leads to accumulation of fat in the liver, and they are having the inflammation or damage to the liver cells. These are the causes of alcoholic fatty acid fatty liver disease is alcoholic hepatitis and alcoholic cirrhosis. Obesity, according to WHO, overweight or obesity is the main is defined as the abnormal or excessive fat accumulation in presence in the body. So, this is a result of the imbalance of calorie taken and calorie absorption or calorie utilization. So, this increased consumption of high calorie food basically required for this obesity and increasing body weight, and decreased level of physical activity increases cholesterol level in the body, which may gives the obesity. So, this is enough for today, for today's lecture. Thank you.